Hello, welcome to our next lesson on forensic science. Starting to use a headset microphone. Hopefully this improves the volume. In any case, we are now looking at Criminalistics, an introduction to forensic science, chapter 11, hairs and fibers. The introduction. Hair is encountered as physical evidence in a wide variety of crimes. It is not yet possible to individualize a hair, human hair, to a single head or body through its morphology. However, partial success for individualizing human hairs has been achieved by isolating and characterizing the DNA to present in hair. When properly collected and submitted to the laboratory accompanied by an adequate number of standard reference samples, hair can provide strong collaborative evidence for placing an individual at a crime scene. Morphology of hair. Hair is an appendage of the skin that grows out of an organ known as the hair follicle. The length of a hair extends from its roots or bulb embedded in the follicle, continues into a shaft, and terminates at a tip end. The shaft is composed of three layers: cuticle, cortex, and medulla. The shaft is subjected to the most intense examination by the forensic scientist. Here you have a cross-section of hair growing out of a tube-like structure called the follicle, embedded in skin. Cuticle is the scale structure covering the exterior of the hair. The scales always point towards the tip of the hair. The scale pattern is useful for species identification. The coronal or crown-like scale pattern resembles a stack of paper cups on top of one another. Spinous or petal-like scales are triangular in shape and protrude from the hair shaft. The imbricate or flattened scale type consists of overlapping scales with narrow margins. Cuticle and cortex. The cortex is the main body of the hair shaft. Its major forensic importance is the fact that it is embedded with the pigment granules that impart their hair with color. The color, shape, and distribution of these granules provide the criminalist with important points of comparison among the hairs of different individuals. The medulla is a cellular column running through the center of the hair. The medullary index measures the diameter of the medulla relative to the diameter of the hair shaft. For humans, the medulla generally occupies less than 1/3 of the diameter of the shaft, while for animals it generally 1/2 or greater. The medulla may be continuous, interrupted, fragmented, or absent. The presence of the medulla varies from individual to individual and even among hairs of a given individual. Medulla also have different shapes depending on the species. Here we have continuous, interrupted, and fragmented patterns of medulla. When it comes to the root and other surrounding cells in the hair follicle, those provide the tools necessary to produce hair and continue its growth. Three phases of hair growth: anagen, the initial phase of growth, follicle actively produces hair; catagen, transition phase; and then the telogen, which is the final growth phase in which the hair naturally falls out of the skin. Hair roots. Here we see top left A, that's the anagen phase. Catagen is B in the top left, and the bottom is C, the telogen phase at 100 times. When pulled from the head, some translucent tissue surrounding the hair shaft near the root may be found. This is called a follicular tag. By using DNA analysis on the follicular tag, the hair may be individualized. Comparing strands, the comparison microscope is an indispensable tool for comparing the morphological characteristics of hair. Scale structure, medullary index, and medullary shape are particularly important in animal hair identification. When comparing strands of human hair, the criminalist is particularly interested in matching the color, length, and diameter. Other important features for comparing human hair are the presence or absence of the medulla, the distribution, shape, and color, intensity of the pigment granules present in the cortex. The most common request is to determine whether or not hair recovered at the crime scene compares to the hair removed from the suspect. However, microscopic hair examinations tend to be subjective and highly dependent on the skills and integrity of the analyst. For this reason, a reported hair comparison must contain DNA information. Some questions worth asking: can the body area from which a hair originated be determined? Normally, yes. Can the racial origin of hair be determined? In some instances, yes. Can the age and sex of an individual be determined from a hair sample? In regards to age, no, unless it's infant hair. And for sex, no. Is it possible to determine if a hair was forcibly removed from the body? Possibly, by examining the hair roots. Are efforts being made to individualize human hair? Yes. Nuclear DNA can be found in hair roots, if present, and mitochondrial DNA can be found in the hair itself. Is it possible to determine whether hair came from a deceased individual at times with certain hairs? Can DNA individualize a human hair? If the DNA is nuclear, yes. If it is mitochondrial, it can distinguish an individual to a maternal line, as in any sons with mothers, or any daughters to their mothers and their mother's mother's mother. That would be the maternal line. Hair and DNA. Recent major breakthroughs in DNA profiling have extended this technology to individualizing human hair. The probability of detecting DNA in hair roots is more likely for hair being examined in its anagen or early growth phase as opposed to its catagen or telogen phases, the middle and final phases. The follicular tag has proven to be a rich source of nuclear DNA associated with hair. Hair and mitochondrial DNA. Mitochondrial DNA, mDNA, can be extracted from the hair shaft. This is found in cellular material located outside of the nucleus of cells and is transmitted only from mother to child. Mitochondrial DNA has a high frequency of occurrence in a population and thus cannot be used to individualize a hair, but it does provide valuable corroboration of a morphological hair comparison. As a rule, all positive microscopical hair comparisons must be confirmed by DNA analysis. Collection and preservation. As a general rule, forensic hair comparisons involve either head hair or pubic hair. The collection of 25 full-length hairs from all areas of the scalp will normally ensure a representative sampling of head hair. A minimum collection of 25 full-length pubic hairs should cover the range of characteristics present in pubic hair. Hair samples are also collected from the victim of suspicious deaths during an autopsy. Types of fibers. Just as hair left at a crime scene can serve as identification, the same logic can be extended to fibers. Fibers can be classified as natural or manufactured. Natural fibers are derived in whole from animal or plant sources, for example, wool, mohair, cashmere, furs, cotton. Man-made fibers are manufactured. Regenerated fibers are manufactured from natural raw materials and include rayon, acetate, and triacetate. Synthetic fibers are produced solely from synthetic chemicals and include nylons, polyesters, and acrylics. Synthetic fibers are composed of polymers or macromolecules, which are a large number of atoms arranged in repeating units known as monomers. Chain-like model of a segment of a polymer molecule. The actual molecule may contain as many as several million monomer units or monomeric units or links. In this case, each individual oval is a monomer molecule component, and when different monomers combine together, they form polymers, polymer chains. Starch and cellulose are natural carbohydrate polymers consisting of large number of repeating units called monomers. Here we have starch in the top and cellulose in the bottom. Looks like it's made out of glucose, that 6-ring sugar. Let's see. And the only difference being the glycosidic bonds and arrangement of how those monomers bond. All these side groups are on the top left versus how they alternate in cellulose. Interesting. Fiber evidence. The quality of the fiber evidence depends on the criminalist's ability to identify the origin of the fiber, or at least be able to narrow the possibilities to a limited number of sources. Obviously, if the examiner is presented with fabrics that can be exactly fitted together at their torn edges, it is a virtual certainty that the fibers were of common origin. Microscopic comparisons between questioned and standard reference fibers are initially undertaken for color and diameter characteristics using a comparison microscope. For other morphological features that could be important in comparing fibers, lengthwise striations on the surface of the fiber, presence of the lustering particles that reduce shine, and cross-sectional shape of the fiber. Here we have different cross-sectional shapes. We have round, trilobal, multi-lobed, flats, and dumbbell. I was not aware of the dumbbell shape. Fascinating. Fiber evidence. Compositional differences may exist in the dyes that were applied to the fibers during the manufacturing process. Methods of fiber comparison. Infrared spectrophotometry is a rapid and reliable method for identifying the generic class of fibers. The molecules that compose a manufactured fiber selectively absorb infrared light to form a characteristic pattern. The microspectrophotometer is a combination of a traditional light microscope and a spectrophotometer. It allows a forensic analyst to view an object under a microscope while simultaneously obtaining its visible and infrared absorption pattern, the visible being the region of light where we see color, and infrared being the higher wavelength, lower energy spectrum of light that we do not see. The visible light microspectrophotometer is a convenient way for analysts to compare the colors of fibers through spectral patterns. A more detailed analysis of the fiber's dye composition can be obtained through a chromatographic separation. Most useful physical property of fibers is that many synthetic fibers exhibit double refraction and birefringence when viewed under a polarizing microscope. Depending on the class of fiber, each polarized plane of light will have a characteristic index of refraction. And lastly, collection of preservation, the investigation tasks of looking for minute strands. Fibers often becomes one of identifying and persevering potential carriers of fiber evidence. Carefully package relevant articles of clothing in separate paper bags If it is necessary to remove a fiber from an object, use clean forceps, place it in a small sheet of paper, fold and label the paper, and place the paper packet inside another container. In any case, just past the 15-minute mark. Glad to have you all. That concludes this latest chapter, chapter 11 on hair and fibers. This will be for forensic science. Enjoying the stuff. Hope you are all learning. Enjoy class. And as always, likes and subscriptions are not necessary, but appreciated just for the heck of it. In any case, peace.